Thank you very much for the very kind introduction and for the invitation to speak here today about our work. As was said, I'm coming from the National Institute of Oncology, where we try to do basic and translational cancer research to better understand the molecular basis of the disease. And we're focusing on cysteine metabolic pathways. And today I would like to give you an overview of some of the projects that are in progress in our laboratory. So cysteine is not an essential amino acid, which means that we can produce cysteine inside our cells from methionine. This is the transsulfuration pathway that you can see on this slide. As you can see, coming from methionine, coming from methionine through homocysteine, you can generate cysteine by consecutive enzymatic reactions. There are two major enzymes that are involved in this process, which is going to be in the focus of my talk today: cystathionine gamma lyase, well, cystathionine beta synthase and cystathionine gamma lyase. These two enzymes utilize in the first reaction step. CBS uses serine and homocysteine to produce cystathionine, and then this cystathionine is used by CSE to generate cysteine. And as you've heard today from the nice talk given by Professor Andreas Papapetropoulos, these enzymes also have secondary functions which generate reactive sulfur species that can induce post-translation modifications on proteins that can affect their functions. And this persulfidation that was introduced to you today by Andreas is going to be in the center of my talk as well. So there will be three different topics on my talk. I will talk to you a little bit about basal-like breast cancer and the role of CBS in this disease. The second part will focus on pancreatic ductal adenocarcinoma. And at the end, I will show you a recently published work how CSE is involved in the development of resistance in BRAF V600E targeted therapy for melanoma. So this first part is focusing on breast cancer, and here we had 45 human breast cancers, and in these 45 human breast cancers, my friend at the Karolinska did a comprehensive protein expression profile analysis, and we took a few proteins which were of central interest to our science that are involved in redox regulation of cellular signaling pathways. And as you can see, there's one protein which stands out in its expression pattern, and that is CBS. Here you can see that it's highly overexpressed. These are basal-like breast cancer tumors, and here is downregulated. These are the luminal type of breast cancers. Basal-like breast cancers are typically triple-negative breast cancers. This is the most aggressive subtype of breast cancers, where treatment options are very limited. Then we looked into copy number aberrations. You could see that there's very little copy number aberrations, and the protein levels correlated very well to the mRNA levels, so we could use the mRNA data sets that we had available by our collaborators, and in many different patient cohorts we could actually show that CBS is always upregulated in basal-like breast cancer. And this talented researcher in my laboratory, Kata Erdei, did very nice work in the biological settings. She was interested whether this overexpression of CBS in this particular tumor type actually has a functional role or not. And she generated xenografts where she knocked down CBS in one cell line, and she had the controls. And as you can see, the CBS knockdowns grew very much slower compared to the wild type of the controls. Cutting the long story short, one of the observations inside these tumors were that in the CBS knockdowns we saw much more necrosis compared to the controls. I don't want to allude into this because we don't have the time, but hypoxia was impaired in this particular tumor type. Angiogenesis was also impaired, in proliferation as well. We have some data trying to explain all this, but focusing on necrosis, at the time of these experiments, this paper came out from a different research group suggesting that basal-like breast cancer tumors are addicted to cysteine, so they really need cysteine to be able to grow. And if they don't have cysteine, then they die a necrotic cell death via this MAC4 NOXA pathway that they proposed. Indeed, we went back to our human tumors and we found that all of those enzymes that are involved in the catabolic pathways of cysteine Cysteine is an amino acid which needs to be very tightly regulated inside the cells, because if the cysteine levels go up, it can be toxic to the cells. And so this pathway, the CDO pathway, is involved in oxidative catabolism of cysteine. This can be highly upregulated to really keep cysteine levels low if necessary. But here you can see that cysteine levels, cysteine is definitely needed in these tumors, because all these catabolic enzymes are downregulated, which means that cysteine is utilized here. Coming to the oxidative stress part, we also confirmed that in these tumors and in these tumor cells you had a higher flux of oxidation. We confirmed it in several different ways. One of the interesting was this PFKB4 upregulation. This enzyme is involved in shunting the glycolytic pathway towards pentose phosphate pathway to produce more NADPH for the reducing machineries, and we demonstrated that this was highly needed in these tumor cells, and other important antioxidants were also highly upregulated, and these cells that were silenced to CBS were much more sensitive to oxidative stress. When we took cysteine away from the medium, of course, what we saw was that they were very sensitive, as published before, to cysteine deprivation, but the CBS knockdowns were more sensitive than the wild types, which makes sense if you think in terms of the fact that CBS is a transsulfuration enzyme which is needed to cysteine production. However, we failed to repeat these experiments that was published before, and in our hands, cysteine deprivation did not induce a necrotic cell death by this pathway. Instead, what we found was that this was a ferroptotic type of cell death that we observed here, and ferrostatin could rescue the cells during cysteine deprivation, and we saw a lot of lipid peroxidation, which is a hallmark of ferroptotic cell death here. And as you can see on the figure, CBS is involved in the production of cysteine, so we thought that if we take serine away from the medium, that will be detrimental for the cells. Instead, we found that serine deprivation together with CBS knockdown could actually rescue this phenotype, which was interesting because it suggested that potentially a secondary function of this enzyme is involved in this biology. And indeed, again, cutting a lot of metabolic analysis short, I cannot show this to you today, but I can talk to you about that in the break if you're interested. What we found was that the secondary function of CBS, which induces persulfidation on protein cysteine residues as well as on glutathione, is very important in this process. The ferroptotic protection was ever since published by Tobias Dick's group in 2023. Glutathione persulfide is involved in rescuing the cells from lipid oxidation. And by knocking down CBS, we saw a significant decrease in glutathione persulfide levels, as well as protein persulfidation was also decreased upon cysteine knockdown. And this was shown to be very important in protecting tile proteins inside cells. This is work from my postdoc in the lab, Eva Doka, who has shown in two elegant papers, we think, that cysteine, when it is persulfidated, is protected from oxidative stress by the fact that overoxidation of these persulfide residues generate these per-thiosulfinic sulfonic acid forms, which then can be reduced back by the thyrodoxine system to the active form. This process not only protects cysteine thiols from oxidative stress, but they also regulate enzymatic functions. As you can see, you can have inactivation of these enzymes by inducing these dynamic post-translational modifications. And we demonstrated it in different systems that it is indeed the case. For example, for PTP1B, we could show that persulfidation is inhibitory, as well as for other phosphatases, which are very important in tumor cell progression. And in tyrosine phosphorylation, we could demonstrate that phosphorylation of PTP1B, P10, and other phosphatases play a major role. Persulfidation of these phosphatases can inhibit their functions. What we found was then when we made single clone cell lines of CBS knockdown, then all those cells that were overexpressing CSE, the other transsulfuration enzyme, were viable. The ones that were not able to overexpress CSE were not viable at the same time. So this suggested that CSE to some extent could substitute for the functions of CBS in this phenotype. And indeed, when we inhibited both enzymes or knocked down one and inhibited the other, tumors were not able to grow in this model system, suggesting that this is indeed important in this particular tumor type. The second part of my talk will focus on pancreatic ductal adenocarcinoma. And one of the interesting messages of this presentation is that this transsulfuration pathway is a plastic pathway. It is utilized by tumor cells in various different ways to promote tumor progression, but it appears that different tumors would utilize this pathway in different ways. In ductal adenocarcinoma, we saw a very different mechanism. What we saw was that, this was done by Agnes Sikora from the lab. We saw that in metastatic tumors, in cells which were isolated from metastatic tumors, CBS was highly upregulated compared to primary tumors which were not metastatic, suggesting that potentially the aggressiveness of this particular tumor type is related to CBS. And indeed, that's what we found in patient samples as well. In the ductal cells, CBS was expressed upon the appearance of metastasis. And we found that in this particular case, CBS was not involved in protection against ferroptosis, was not involved in angiogenesis or in the hypoxic response, but it was involved in the aggressiveness of the tumor and in the metastasis formation, as we saw with this animal model, as orthotopic injection initiated animal model. Regarding the mechanism, we demonstrated that this was due to the epithelial to mesenchymal transformation. So CBS promotes epithelial to mesenchymal transformation. And again, to cut the long story short, we've shown that it is via STAT3, the transcription factor, which cannot be phosphorylated if you knock down CBS due to its oxidative modifications, which is protected in the presence of CBS by persulfidation-related mechanisms. But I would like to spend a little bit more time on the third part of my talk, which is related to targeted therapy of melanoma, BRAF V600E targeted therapy, which is the first targeted therapy in this particular disease. And in particular, we did a study how this drug, what is the underlying mechanism of the resistance development in this drug. So this is BRAF, and this mutation you can find in about 50% of the patients. This V600E mutation is up here. And this is an important mutation because, as you can see it on this slide, if you have this mutation in patients, BRAF is constitutively activated. The way it works, it has a cysteine or a glycine-rich loop at the active site, which opens up upon RAS-mediated activation. But if you have a V600E mutation up here, then it's always open, so the enzyme is constitutively activated, which results in phosphorylation and cancer development. And there was this drug which was introduced to the clinic about 2 decades ago, vemurafenib, which was the first targeted therapy for this particular disease. And that drug was extremely effective initially, and you can see that in a stage 4 cancer patient, the drug response was very good. But unfortunately, the disease came back in a more severe form, and the resistance developed in this particular case and in apparently all cases. And what happened was that people started to try to understand how the resistance develops for this particular therapy, and it appears that you can activate MEK by different pathways. I don't have time to allude into that. And then they introduced combination therapies. This dabrafenib trametinib therapy is now used in the clinic. However, as you can see it down here, this combination therapy can only give patients the additional 3 to 4 months in the progression-free survival. So resistance development is still a big problem in this particular disease, and we wanted to understand a little bit more about how this resistance developed. And this was done by this talented PhD student, Claudia, in the lab. We just published this work in May. And she first generated cell lines which were resistant to dabrafenib trametinib treatment, and she had this model system where she first had the drug-naive cells, and then she added the drug to the cells and looked into gene expressions at different time points. Then she had the resistant cells, and she took the drug away and saw what happened there. And she confirmed that indeed the previously published results were true. The PI3 kinase pathway is very important to take over signaling when the resistance develops, but I will not go into those details. Instead, what I will show you is that our analysis demonstrated that upon dabrafenib/trametinib treatment of these cells, you induce a large oxidative stress inside the cellular environment. We could demonstrate that on the overoxidation of peroxyredoxins, which are the redox sentinels of the cell, as well as on DNA damage upon additional oxidative stress to these cells. And we also captured intermediate oxidative forms of cysteine residues on proteins. We show that this is done mostly by mitochondrial respiration, because there's an elevated mitochondrial respiration upon drug treatment in these cells, which as a side effect generate more superoxide, as well as by cytochrome P450 enzymes, which are upregulated in these cells upon drug treatment. It is a really long story, but what I need to tell you is that looking at all these antioxidant genes, we found that they were all upregulated upon drug treatment, so they are all involved in the antioxidant protection against the induced oxidative stress in these cell lines. Again, the PFKB3, PFKB4 pathway is involved in generating more NADPH, which is required for the thyroidexin and the glutathione system to reduce the oxidative stress, induced modifications on proteins, and many other factors demonstrated and underlined a severe oxidative response upon drug treatment inside these cells. So how the cells, those cells that can survive the drug treatment, we call them persister cells, can indeed survive this environment. And one interesting finding was that this channel, this antiporter, cysteine glutamate antiporter, was highly upregulated in these cells, suggesting that these cells indeed need more cysteine, potentially to overcome oxidative stress. And the uptake of cysteine, this is the disulfide form, which is transported inside the cells from the extracellular space via this channel, is indeed up in these cells. So you have more cysteine transport upon drug treatment into these cells. And when you block this channel, then you see diminished development of resistance. So cysteine is very important for these cells to survive the persister cells upon the drug treatment. But what was really interesting and puzzling at first was, if cysteine is so necessary and needed for these cells to survive, why do you see less cysteine in the intracellular space? We could not find a good explanation for that initially, but apparently, if you think about it, it would also underlie the utilization of cysteine potentially via these other pathways where cysteine is needed, for example, to produce glutathione. When we looked into the glutathione production, indeed we found that a number of proteins that utilize glutathione for protecting the cell, GPX1, GPX4, and GST-pi. First two is involved in protection against oxidative stress, and GST-pi is an enzyme which is involved in clearing the xenobiotic, the drug. They're all up, and they use glutathione, and glutathione production is indeed highly upregulated inside these cells. So one pathway of cysteine consumption is to make more glutathione in this system. But another interesting, well, first I talked to you about the glutaminolysis, which also suggested that indeed this glutamate cysteine antiporter is involved in protection, is that glutaminolysis is also highly upregulated. So you produce, the cells take up more glutamine and produce more glutamate, which is not only utilized for the antiporter, but also for glutathione synthesis. That was nice to see, but what was really puzzling again is that we saw a huge upregulation of the cysteine catabolic pathways. So why would the cell destroy the cysteine if it needs it so badly from the extracellular space via the XCT? But what is important to understand here is that the antiporter actually transports cysteine, not cysteine. So perhaps it's the cysteine that is needed for the cells, not the cysteine. That was the working hypothesis then. And another important observation was here that upon drug treatment, this enzyme CSE was immediately highly upregulated. CBS levels went down, and then when resistance developed, CBS levels came back and CSE went away, suggesting that it's not cysteine production that is important, but potentially the secondary functions of these enzymes. And that is exactly what we found. And what was interesting is that these enzymes, they can also use cysteine, the disulfide form of cysteine, as substrate to directly generate cysteine persulfide. And this is indeed what happens. This is a better substrate compared to cysteine, but in normal conditions, it's barely ever a pathway that operates inside the cytosol because of the reducing environment. But upon cysteine consumption and elevated cysteine transport, you can generate conditions where fluxomic and metabolomic analysis, this is also again a lot of work, demonstrated clearly that this was the case. So cysteine was directly utilized by these enzymes to produce persulfide species. Why are these persulfides important? Oh, first we did transfection of CSE and then knocked it out from cells to show that it's indeed CSE that was involved in the production of these reactive sulfur species, namely the cysteine persulfides, and indeed that was the case. Then, of course, to make sure that it's not some side effects of working with cells, we used xenografts and patient-derived xenografts and demonstrated that the metabolomics were the same as we observed it inside cells. But mechanistically, what was interesting to see is that when we silenced ETHE, which is also a protein persulfide dioxygenase, so involved in catabolic pathways of persulfides, then cells were much better off and developed resistance faster. So persulfides are good for these cells for tumor progression, and in particular persister cells, we believe, utilize these persulfides that are generated by CSE for survival in one way or another. And mechanistically, again, we were able to demonstrate that one mechanism was to protect important cysteine residues from overoxidation. This was demonstrated by a small proteomic analysis and also in peroxyredoxin 2 and some other enzymes. This was the case. But another interesting aspect of this cysteine persulfide production was that we could also show that the electron transport chain in the mitochondria utilized hydrogen sulfide and cysteine persulfide as an electron source to produce thiosulfate via the SQR machinery, and also it utilized the citric acid cycle and also glutaminolysis to give more spare respiratory capacity for this electron transport chain, and these two together generated more ATP, which was then utilized by ABCG transporters to pump out the drug, which was another interesting aspect of persulfide utilization in persister cells. It was really difficult to show it and demonstrate it in patient samples, because when patients are still under treatment and the drug is still effective, you don't really take samples from these patients. But we managed to find 2 patients where from the primary tumors, as well as 2 patients where from the metastasis the samples were taken, and indeed CSE in these patients were highly upregulated. When the resistance develops, CSE goes back down, so you don't see this anymore in patient samples. So this was really a challenge, but we managed to show that this is happening. And what is most interesting here is that if you pharmacologically or genetically inhibit CSE in this model system in the cells, you can actually inhibit resistance development very efficiently, suggesting that this could be an interesting combination therapy for these patients. And what was also required to show was that in animal models, when we utilized a CSE inhibitor, propargyl glycine, together with the drug, you can see very efficiently progression-free survival was elongated in the combination therapy. So suggesting that if you use a CSE inhibitor together with these drugs, then drug resistance can be inhibited to a large extent. We think that in human models this could indeed be more efficient than what you see here because of the immune system, which also has a major role, but I don't have the time to go into that. But I think with that I wanted to thank the team. It's a very talented group of individuals who did the work. We have a number of collaborators who were involved. And of course, I'm very grateful for the organizers, for the invitation, and for the funding for some funding agencies. Thank you for the attention.